everyone. My name is Daniel Wickland, and I'm an assistant professor of biomedical informatics at Mayo Clinic in Jacksonville, Florida. Today, my talk is going to focus on racial disparities in genomic data quality in cancer. This is a study that we published last year in the Journal of the National Cancer Institute. To start off, in the United States, cancer disproportionately impacts African Americans. For example, African American women have higher rates of aggressive breast cancer, and African American men have higher incidence and higher mortality rates of prostate cancer compared to Caucasian men. Identifying the factors that underlie these cancer disparities requires data that are equitable in both quantity and quality across racial groups, but differences in genomic data quality between racial groups have not been well studied. We studied data from the Cancer Genome Atlas, or TCGA, which is one of the largest and most widely used cancer sequencing projects. It contains data from more than 30 different cancer types, and 7 of these cancer types included at least 50 self-reported Black patients. Those cancer types are breast cancer, prostate, lung, uterine, colon, and 2 types of kidney cancer. In TCGA, the racial designations are based on self-report. But in order to have a more consistent approach to assigning racial designations, we used principal components analysis that was based on comparing the genotypes in TCGA with the genotypes in reference samples from 1,000 genomes that have known pure ancestry. So we focused on the Black patients from TCGA and the pure African samples from 1,000 genomes, and also the self-reported white samples from TCGA and the pure European samples from 1,000 genomes. There are other racial categories in TCGA, but we decided just to focus on these 2 because they had by far the biggest cohort sizes. In most cases, our PCA approach classified ancestry in the same way as self-report. For example, in breast cancer, there were 169 self-reported Black patients in TCGA, and our PCA approach inferred African ancestry in all 169 of those. For the self-reported white exome samples in TCGA, we identified not only all 642 of those as being as having European ancestry, but we were also able to rescue an additional 58 samples that did not have any self-report information in TCGA using this approach. So over the course of just routine QC and data analysis, we made the totally incidental finding that in TCGA, exomes with African ancestry had lower overall read depth compared to exomes with European ancestry. This plot shows on the top panel primary tumor, and the bottom panel is patient germline. Again, this is the overall read depth per exome, so every data point is 1 individual. And the units are in millions of reads per exome. So just using breast cancer again as an example, the average or the mean read depth among ancestrally African exomes is 90.4 million reads, whereas in European exomes it was almost 120 million. And this is a highly significant difference. And we found similar differences in 6 of the 7 cancer types that we investigated. And again, this is an observation that was consistent in both tumor and germline data. As I said before, we're just focusing here on African and European ancestry due to cohort size. So this figure shows the total number of reads per exome. We also looked at just the total number of mapped reads per exome. And the observation persisted. There's significantly lower overall read depth among African exomes compared to European. And the data is not shown here, but we also looked at unmapped reads and saw the same thing. So we hypothesized that perhaps the difference in overall read depth could be due to tumor purity, because in cancer sequencing experiments, if tumor purity is low, meaning that the ratio of tumor to normal tissue in the sample is low, then often read depth, samples will be sequenced at a greater read depth in order to obtain more tumor supporting reads. So one possibility here is that the European samples may have had lower tumor purity and consequently were sequenced at a higher depth to try to compensate for that. But that was not the case. We compared tumor purity between the African exomes and the European exomes, and there was no difference. Tumor purity was essentially the same in both of these groups. So that led us to think of other possibilities, other factors that might explain why ancestrally African individuals had lower read depth in TCGA. Next, we looked at the different sequencing centers, because it's possible that one sequencing center was consistently, systematically sequencing at a lower depth than the others. But the data don't support that idea. There were 3 sequencing centers here for the 6 different cancer types. Center 1 sequenced the prostate and lung cancer samples. Center 2 sequenced the breast cancer and uterine cancer samples. And Center 3 sequenced the colon and kidney cancer samples. And we can see here that even within a given center, the racial disparity is persisting. For both tumor samples and for germline samples, African exomes have lower overall depth than European exomes. So this indicated to us that differences between sequencing centers could not explain the racial disparity. Next, we looked at the different sample collection sites. For the cancer types that we're investigating here, there were 13 or 14 different sample collection sites. And we thought maybe some of these sites just happened to, maybe they sequenced at a lower depth. Maybe something was different about the sample collection protocols that led to differences in overall depth between the different individuals, but we didn't see that here. For almost every comparison here, the African samples, again, had lower read depth than the European samples. There were a few cases where the difference is no longer significant. For example, at site 1, I know it's a little hard to see, but there's really no significant difference between African and European, but you can still see that visually the distribution is more towards the lower end of read depth among the African individuals than the European. So even if it's not significant, the trend was generally the same. So this indicated to us that sample collection site was also not responsible. Differences between sample collection sites were also not responsible for the racial disparity in overall depth. So we knew that there were a few other factors, a few other experimental conditions that varied between the different samples. The next one that we looked at was exome capture kit. Exome capture kit is used to enrich targeted regions of the genome during library preparation. And this is what allows us to sequence only the exome instead of the entire genome. For this capture kit analysis, we focused only on the breast cancer data set because the largest number of African exomes, as well as the biggest racial disparities in depth, could be found in this data set. For our study, there were 3 different capture kits that we looked at. Although the TCGA breast cancer did use more than just 3, we were only able to find detailed information about the capture regions for these kits, for these 3. All 3 of them are Nimblegen kits. There's the Nimblegen HG18 capture kit, Nimblegen SeqCap Easy Exome version 2, and CCAP easy exome version 3. So if we restrict our analysis only to the samples that were processed using these Nimblegen kits, in the first panel here on the left, you can see that there's still a very obvious racial disparity between the African and the European exomes, with lower depth in the African exomes. But if we look individually within each capture kit, there is no difference between the African and the European samples anymore. For example, in the HG18 exome version 2 kit, there were 38 African exomes processed by this kit and 380 European exomes processed by this kit. And they were all sequenced at essentially the same depth. There's no significant difference anymore between the African and the European samples. And there's a similar story for the other two kits. But the reason that African samples had lower overall depth is because the majority of African samples were processed using this version 3 kit, and all of the samples processed using this kit, including the European, were sequenced at a lower depth. Whereas the HG18 kit, which was used to sequence the majority of European samples, read depth is substantially higher for both African and European exomes. We don't know the exact reason why these batches were sequenced at different depths, but regardless of the reason, this is the explanation for why we're seeing systematically lower overall read depths in the African exomes as a group. In addition to the breast cancer data set, we also inspected the 6 other cancer types. In some cases, we found similar results. In some cases, we didn't. So in uterine cancer and kidney cancer, once again, the European samples were enriched in batches that are processed by these earlier capture kits. And I describe them as earlier because, for example, this kit, the HG18, this kit was developed when the HG18 version of the human reference genome was in vogue. These other kits, the version 2 and the version 3, were used or were manufactured when the HG19 version of the genome was more widely used. So these earlier kits were samples processed by these earlier kits were sequenced at a higher depth. For the uterine data set, as well as the kidney cancer data set here, 3 other cancer types, prostate, lung, and colon, the racial disparity was still observable, even if we separate the samples by kit. Although for 2 of these cancer types, only 1 kit was actually used. And we couldn't find very detailed information about the kit, about the capture region. So we didn't pursue any further those cancer types. Instead, continued our focus on breast and uterine and kidney. So we showed that there's a difference in overall read depth between African and European exomes, but a bigger question is, does that actually matter? So next we investigated the impact of that lower coverage on germline variants discovery. Because TCGA only reported the somatic mutations, we took the germline BAM files from TCGA and performed multi-sample genotyping ourselves using GATK. We also, just for variant quality control, we used the variant quality score recalibration method in GATK, and again focused on the breast cancer data set because of its larger cohort size and most significant racial differences. We knew from previous studies that the sensitivity for variant detection declines significantly once read depth falls below 10 reads. And that's shown in this plot on the left. At various false positive rates, once read depth is less than 10, sensitivity dramatically goes down. So we asked the question, are genomic positions that have coverage below 10 reads overrepresented among African exomes. So here, rather than focusing on the overall depth per individual, we're focusing on individual positions in each individual. This series of plots is a set of mosaic plots that show enrichment of racial groups in different coverage categories. In this one on the left, the Nimblegen exome HG18 version 2 kit, there's an overrepresentation, which is indicated by the blue color, overrepresentation of African individuals at positions that are covered by 1 to 10 reads and 11 to 39 reads. In contrast, at coverage of more than 40 reads per position, there's an underrepresentation of African individuals and an overrepresentation of European exomes. And that observation is consistent across these different kits. So the important point here is that even though the overall sequencing depths within each kit do not differ between African and European exomes, which is what I showed a few minutes ago, African exomes still have a higher than expected percentage of genomic positions with low coverage in all 3 of these kits. So that suggested to us that variant detection could be impaired in the African exomes compared to the European. Next, we looked at the minor allele frequency within the common capture region of those kits. This figure shows the minor allele frequency in the TCGA breast cancer cohort on the y-axis, and the minor allele frequency in the 1,000 genomes populations on the x-axis. The different colors here just indicate the different coverage levels, and the solid lines are for the European samples, and the dotted lines are for African. So we can see that at higher coverage levels, which are shown here in the blue and the black lines, the minor allele frequencies are very, very similar between the breast cancer cohort and the 1,000 genomes cohort. And that's indicated by the line here, the lines that have slopes of almost 1. So a minor allele frequency of 0.6 in 1,000 genomes corresponds pretty well to a minor allele frequency of 0.6 in TCGA. And I'll try to highlight that here with this pen. So if we go up here, minor allele frequency of 0.6 in 1,000 genomes, minor allele frequency of 0.6 in TCGA. However, at lower coverage levels, such as at 10 reads or fewer, or 5 reads or fewer, and that's shown by the lines here in orange and purple, there's an under-detection of germline variants in TCGA compared to 1,000 genomes. For example, at again, using the read depth of 0.6 in 1,000 genomes, well, if we look specifically at samples of African ancestry at coverage below 5, the corresponding minor allele frequency here in TCGA is about 0.25. So that's indicating to us that there's systematic underdetection of African variants at low coverage positions relative to European. Next, just focusing on the 1,000 genomes data, we compared the minor allele frequencies within the common capture region between the African individuals and the European individuals. And we found that the target capture regions are more polymorphic in the African individuals, which is shown here. That indicated to us that the capture probes are likely to be less effective for African exomes. Presumably they were designed based on European exomes. Next, we looked at the impact of lower coverage on somatic variants discovery, and we had very similar findings, although the trends were a little bit less consistent. But I would draw your attention here to the capture kit on the right side, this Nimblegen CCAP Easy Exome version 3. This is the kit that sequenced the majority of African exomes, 123 out of 172. And in this kit, there is an overrepresentation of African individuals at positions that are covered by fewer than 40 reads. Very similar finding to what we saw in the germline variants. This is a plot just showing the number of reads supporting the somatic variant alternative allele across all somatic variants in this data set. So you can see here that TCGA really made every effort to report all somatic mutations, even if they were covered by just 1 read or 2 reads or 3 reads. Even with high sensitivity mutation culling, many somatic mutations will still be missed at low coverage positions. And that's an idea that this series of plots is attempting to convey. So if you look at the bottom panel here at 5 times coverage, in all 3 of these plots, the distribution here is the distribution of B allele concentration, which is, it's a fraction. It's the number of reads supporting the somatic variant divided by the total number of reads at that position in a given sample. So if we assume 5 times coverage, that means on average 5 reads are supporting any given position. And if we also require only 1 read to support the somatic variant, that means that the minimum B allele concentration that we can detect is 0.2, because 1 read out of 5 reads is 0.2. But in TCGA, breast cancer, over half of the variants had a B allele concentration below 2. So if we only have 5 times coverage, we would be missing all of those variants. If coverage is increased to 10 times, then the minimum B allele concentration that we can detect is 0.1, because 1 read out of 10 reads is 0.1. In this case, we'd be potentially missing 22% of the variants in this data set. And of course, at higher coverage levels, we're missing very little. So the take-home message of this figure is that low coverage positions, which we know are overrepresented among African exomes, low coverage positions can only report mutations with a high B allele concentration. So we're potentially missing out on a lot of variants, especially in the African samples. To summarize, we found lower overall coverage of both germline and tumor exomes from ancestrally African patients in TCGA. This disparity in overall coverage and overall depth was not due to tumor purity differences between African and European exomes. It was also not due to sequencing center or sample collection site. Instead, it was due to the imbalanced allocations of African and European exomes among sample batches that were sequenced at different depths by different capture kits. In terms of capture kit design, we found that African exomes have more polymorphisms in the targeted regions. So there are really 2 explanations for the differences in read depth. We have the lower overall depth, as well as disproportionately higher number of positions with insufficient coverage in patients with African ancestry. So those 2 factors led to undercalling of germline variants and somatic mutations in the Cancer Genome Atlas. Well, that concludes my presentation. Thank you very much for your attention.